Good evening, ladies and gentlemen. It gives me great pleasure and privilege to announce Dr. Tusit Weerasooriya, who's going to give us a talk on quantitative visualization and modeling of biological material. Before he starts, let me give a small background information about him. Tusit got his bachelor's degree in University of Sri Lanka with a first class in mechanical engineering. Then he went to the University of Cambridge, England, where he got his PhD. Excuse me. Since then, he's been working for the US Army Research Laboratories and been a project lead in his area of expertise. With that, I give you Dr. Tusit Weerasooriya.
Thank you, Chandru. I work for Army Research Lab, and I'm kind of lead multi-scale mechanics research in soldier protection sciences branch. My background is in mechanical engineering and after that material science. And I have been working starting from metals to ceramics to polymer, hard polymer, soft polymer, and now working on biological material and different length scale from cellular to tissues to components. So my presentation today is quantitative visualization of and modeling of biological material. When I say quantitative visualization, mean I try to visualize the how material deform and fail, and then quantify from that information, get stresses and strains, and try to relate them and come out with models so people can use that in computational framework. For example, in our case, I'll talk to you about a helmet. So a helmet is, so when I have a blast load explosion or impact on the helmet, there will be a load transfer from helmet to the pad, to the skull, to the brain. So we are interested in optimizing the helmet geometry, material, pads, etc., to reduce the injury to the brain. So that is where we need cellular level injury mechanisms. Still, there is a long way to go to get those models. But I'm now working on skull. So we can use skull as an injury criteria, skull fracture. And then use that to optimize my helmet and pad so there won't be a skull fracture. So that's all right now we have is being able to quantify skull fracture for head injury. So also I worked a few years ago on human femur. There's some nice work were done, and all those are published. So you can, I have references for those, so you can look at the references to learn more detailed information from that work. So outline for my presentation is cortical femur mechanical response. Then I will introduce you for getting fracture behavior and quantify. I don't know how much you have information, how much you know about fracture mechanics. Fracture mechanics is trying to parameterize stress field around the crack tip. So that parameterization is called stress intensity factor. So this K1 is in one direction, tensile direction, and K2 in shear direction. So we will show you how we get that through optical measurements. And so also I will show you some deformation experiments we have done on cortical bone in two directions. Cortical femur bone is directional, transversely isotropic. So far, what we have in literature is assuming bone is isotropic. So this is the first set of data done on especially from cadaveric bones. And we have a new data set showing the orientation effect. Also, I specialized in my last 30 years developing high rate behavior of material that is very relevant to the Army. Army, we have things impacting, penetrating, all that happen at very high rate. So it is very important for Army to understand how material deform and fail at high rate. And then we have this challenge of having to look at all classes of material, ceramics. That's how I moved from metal upward to biological. Ceramic, then even I have looked at single fiber, micron scale fiber, trying to get or getting deformation and failure response. So we have at ARL, real nice lab we have developed to obtain high rate behavior of all different kinds of material. And second part of my presentation is skull. We have done a lot of work on skull. We have looked at human skull and then also animal skull. And animal skull is mini pig. We have been using mini pig as a surrogate. But we can conduct experiments on mini pig and get injury criteria. But we cannot do similar things on human. So we are looking at how we can transition what we get from mini pig studies to human studies. So that's one of our big challenge. And then I will show you what we have done on compressive response of skull material. And we have developed unique modeling capability relating the compressive response to microstructural detail. And recently we have developed some validation experiments, what I call high rate indentation experiments. We are trying to indent at high rate the skulls and trying to get measurements so that we can validate our what we get from computer codes. So objective is to all our studies develop methodologies to obtain microstructural dependent and constitutive and failure modeling strategies for bones. So, so I already talked about motivation to relate for skull work, relate brain injury to high rate mechanical loading. And so we need to know pressure and deviatoric strain tensor that lead to the injury. So first I will talk about our femur work. We have got femur material from 3 donors, from 36, 43, and 50-year-old subjects. And we have looked at fracture, rate effects, mode mixity, whether it failed from shear or tensile loading. Please interrupt and ask questions. I may be introducing some words which I will try to give if you are not following, I'll try to give a brief introduction for those words. Already I talked about femur rate and orientation dependent studies we were doing, then microstructural effect. These are preliminary work. A lot of these things I already mentioned. So femur is transversely isotropic material. So this is a section this way. So it consists of tubular structures called osteons, and they are aligned this way. So what? This is some micro CT work we did on femur. You can see all these canals are aligned in the long bone direction. So what we did was we got these femurs from donors, and then we got fracture specimens. These are small beams, ASTM standard beams. And so we had obtained these specimens in 4 directions, front, back, and 2 sides. And we had 6 femurs. So when we get this, we sanded them, and then we inserted a micro crack into it, micron level crack with a diamond saw. So if you are into material science, you probably know something called 4-point bend fracture specimens. These are ASTM standard. So what we did over here is this is the beam, this is the crack. We have 4 points, and then we push them and bend them, and then get the crack to initiate and grow. So that's the kind of fracture experiments we did. Okay. Yeah, it's a pre-crack. Pre-crack was introduced with a diamond saw, because these are bone specimens, so that's the sharpest we could get. Okay. So it's about 100 micron slit. Okay.
When you are making your specimen ready for a specimen preparation, you are cutting it from the whole ball.
Yeah.
Introduce any protection when you are cutting it?
Initially, there may have been some injury, but then we sanded that away with the fine sand. So but also introducing this crack, that's notch, that's where action is going to be. So any damage over here will not affect the bending specimens. Okay. So one thing we did was, in our lab, my lab, I try to look at the rate effect. That's our specialty. So we're trying to get high rate effect, how things fracture at very high rate, like blast loading and that kind of rate. So we have developed a high rate fracture method. I don't know whether you know, the high rate loading is, there's a technique called Hopkinson-Bart method that some seem to already know. So we use that method, but then we have developed a high rate fracture method to get fracture initiation at high rate. So this is the reference if you want to look at. And other thing we did was, this is the attachment we developed. So this is the specimen over there, and these are the points, these 4 points. So we push this at high rate through impact. And then one other thing we did was, one unique thing we did was, we used very high-speed cameras, 1 million frame per second camera, and looked at the crack tip, how crack initiates and propagates. So the details of this method can be seen in this. You can get details from this reference. So this is an experiment that you can see the crack is initiating and growing. And this is the microsecond timer. So each microsecond we were taking a picture. In addition, another unique thing we did was we speckled these specimens with a very fine paint, speckling. And from that, tracking these speckles, how they move through optical observation, we can get the displacements, what we call U and V, X and Y direction. From that, we can get these displacement maps, how crack initiates and grow. And then at the crack tip, we can get the displacement and also strain field. So we can get the strain to initiation, displacement to initiation at very high speeds. Also, what we did was, first, as everyone else, we assume this material is isotropic. So for isotropic, we have these stress intensity factor relationships. From the dimension of the specimen, you can get these stress intensity factors. So this is a slower rate, this is a function of loading rate, intermediate rate, and these are high rate Hopkinson bar data for crack initiation. When crack initiates, we can get the load, and from load with this expression, we can get the stress intensity factor for crack initiation. This is the isotropic stress intensity factor for crack initiation. It's a tensile, we assume it's tensile crack initiation. So though crack move in perpendicular to this notch direction, we initially assume it is going to be mode 1, what we call tensile crack. But due to this anisotropic, if this material was not transversely isotropic, they are aligned this way, those tubes. That is why crack is deviating. So we assume this material is isotropic, but that is not right. So neat thing over here is at the crack tip, we get U and V field, and then you can see it's not symmetric. So it contains both shear as well as tensile data. So next step is to how we can use this to get what we call K1 and K2, tensile and shear stress intensity factors. So before doing that, what we did was traditional way. We got stress intensity factor at different loading rates and plotted. So these are for 3 donors. So there is no statistical difference between each donor for the initiation of the crack stress intensity factor. So this is a slower rate, then it increases, then it plateaus at high rate. Also, what we observed was for lower rate, crack always turn 90 degrees. It becomes what we call mode 2 cracks. Whereas other rates, it is more mixed crack. It was 45-degree crack initiation. So that is, so these tubes are aligned this way. So crack to grow and turn 90 degree, it's an easier path. That's why stress intensity is lower for crack initiation. Also, this is looking at as a function of age. We did not see any difference. I would really like to look at younger and older. There should be a difference in this. But we were not able to get donor specimens. Also, this is a comparison of human data with animal data, which is in literature. So they have the same trend, but all are assuming isotropic models, isotropic representation. Also, when we are doing finite element analysis, we want to load the specimen, and at a certain stage, we want to say, here, crack initiates. So we need criteria. So one is this stress intensity factor criterion. Other one is because we are measuring crack tip information, So we can get the maximum tensile strain here for crack initiation. So this is maximum tensile strain plotted. So if someone is doing finite element analysis, so they can track tensile strain, and it reaches this critical value. You can say crack initiates in the bone. Other criteria people use is energy criteria. So we got that information also. But the neatest thing is next, trying to get anisotropy into this. And because we have all these optical measurements at the crack tip, displacement at the crack tip. This is the notation we use if you are not familiar. This is the crack. This x direction, y direction. Displacements are called u and v. U is the displacement in x direction. Y is the displacement in the v is the displacement in the y direction at a r theta coordinate. So mode 1 fracture is crack opens, tensile. So this is an example of mode 1. Mode 2 fracture is shear fracture. When I use those terminology, that's what it means. Also, we have this relationship, U relating to R and theta, and then stress intensity factor, K1 is the mode 1 fracture, and K2 mode 2 fracture. So with these relationships, we know for each R and theta, U and V from our measurements, optical measurements. And so with these equations, we can get, when we know UV relationship at the crack initiation, we can get K1 and K2. Here, so using this method for lower rate, high rate, intermediate rate, and high rate, we got K1 and K2. Please ask questions if you're not following this. So, yes.
Testing.
Here, can we use the microphone?
For your split Hopkinson pressure bars testing, are you running the test in compression mode or in tension mode, knowing that I would think that the crack growth would be more critical in tension rather than compression?
So if my specimen is like that, I'm bending like this. So it's in a compression mode. Bar is pushing it. Bar is in compression mode. But due to bending, at the crack tip, you have a tensile strength.
Okay. So it's not like a uniaxial Hopkinson bar testing?
No, this is not a uniaxial standard Hopkinson bar test. This is a bend test. This is a modified. I'll show you later some. We did compression test also. So we are using Hopkinson bar to generate, do bend test. So that's that paper which we developed that method. So you can look at that. Okay.
Yeah, over here. So what kind of instrumentation are you using? And this is, in the lab in vitro, and you don't take into account effects, with the connective tissue between, at the femur and knee joint or hip joint, and I was wondering. So this is just on plain cadaver bone that's relatively fresh. Okay, so what type of instrumentation are you using to measure the different parameters?
Different parameters means for like connective tissues?
Yeah.
So right now...
just before the fracture occurs.
So before fracture occurs, it deforms.
Yeah.
Right now, with this study, we are looking at fracture initiation, this bend test, only looking at how fracture occurs. I will show you later. Also, I have done a compression study, uniaxial compression study, where I'm getting data so I can get, or I, working with universities, can get models to represent the deformation, what we call constitutive. So in actual loading, these are ideal material testing to get these parameters separate, compression. So in real case, your bone deforms initially. So when I have the models from compression test, I can use what we call constitutive models. So that we will put into the code that will allow me to deform the bone in the computer. But I have to tell to the computer somewhere, oh, there's enough, it bent, that now it should fracture. So this is getting fracture criteria. For computer codes to work, there are two things, deformation and fracture, which we call constitutive model and failure model. So this is to try to get the failure model. Somewhere you have to, otherwise it will keep on deforming. So, but in reality, we need to tell that if you exceed a critical value, so that's what I'm looking for, what value to tell. So it's a strain, maximum strain, energy, stress intensity factor. So those are the things I'm studying now to get those criteria and put into the codes.
Okay. So I want to make a couple of comments about the fracture pattern. We also done some. Right. So we have also done some testing on the femur, and the fracture pattern on the whole femur, when you put a bending, it's actually the tensile part of the bending has a fracture perpendicular to the bend in tensile strain, and then on compressive side in bending, the other side is the compressive side. You actually have some kind of butterfly pattern. So the fracture line of plane is actually almost like 45 degrees, as you've seen it. So in comparison to your test specimen, you essentially have a pre-cracked a line of plane that's simulating a tensile part of the fracture, which would have occurred if you tested it without a pre-crack. And then you see those strain pattern from DIC that it's 45 degrees. Okay. That seems to be indicating that in compression, the fracture usually occurs 45 degrees to the compressive side. Does that make sense to you?
Yes. So I'm looking at tensile fracture criteria right now. I don't think anyone in the world right now can get the butterfly and the full fracture. I don't think even metal we can get full failure models predicting the fracture patterns. So I'm trying to focus right now on initiation of the fracture, okay? Because we can do in the lab all these experiments and say this kind of fracture occurs, but in the end, we have to put this in code so we can use this to design things. Right now, we cannot determine all different complex fracture. As soon as fracture occurs, whole stress field changes. So we are not in that stage yet.
Okay.
So there's so one mode is tension. The other one, this can crush in the compression. So that I'm not looking at because tensile mode is more critical.
Yeah, I don't understand. Tensile in human bone, the strength in tension is actually weaker than in compression for human femoral bone. That's the findings that in biomechanics literature.
So what you're saying is when I get a compression specimen and then compress it and then it fails somewhere. So that Yeah, I'll show you there.
If
We have done compression experiments also.
Okay, thank you.
So how much time?
10 minutes.
Only 10 minutes left?
15 minutes.
Oh, okay. I need to show my skull work. So
Since you got all the questions, so 15 minutes.
Okay. So what we have done over here is from this K1, because I was able to determine K1 and K2, so I can prepare what I call K2 mode mixity maps. And then these are all my data points. These are the field data points are crack growing 90 degrees, and the other one are 45 degrees, this unfilled. So I was able to get a K1 threshold for crack initiation, and neat thing is I was able to get a K2 threshold above that where crack could go 90 degrees. So this is for slower rate. Most are 90-degree cracks. Then as strain rate is increased, so the less mode 2 cracks. So still I have a threshold, but it increased. And then higher rate, Hopkinson bar rate, all are mixed mode cracks. So all are 45-degree cracks. So these are the compression work we have done, you are asking. So at different rate, 3 rates, slow, intermediate, and high rate, and in 2 directions, this direction and this direction. So the failure stress in this case for compression is, so it just go flatten. So this is the compressive stress for failure for bone in 2 directions for femur. So there is a paper, all these you can read. So skull work is really nice and more complex compared to femur. So when we do a skull experiment, we do a clinical scan before, and then that is about over 600 micron. So you don't see the skull structure. It is like a more continuous structure. So people just use one modulus for the whole thing, but it is not right. So this is for animal and human. So these are the structures we have done micro-CT on. So one type of a mini pig, and it's what I call functionally gradient, porous structure changes. And this type of mini pig, we found porosity does not change as a function of thickness. Whereas human, so you have look like a hard shell, outer layer and inner layer, and then middle is a porous structure. So first thing is we have these different microstructures. So how do we quantify that? If I want to get models of modulus as a function of microstructure, so each location microstructure is different. So first challenge is how do we quantify the microstructure? So we looked at this is the mini pig, and we looked at how porosity changes, pore size, average pore size changes as a function of thickness, and then wall thickness, how it changes.
Don't touch this on the screen.
What is that?
Don't touch it on the screen.
Okay. So, and also we looked at how these pores are aligned, what we call mill tensor, to see whether there is any anisotropy. And we found that porosity is the governing factor for modulus. And this is for human, how porosity changes, even though it looks like outer layer is a hard layer, and it does have some porosity. So we have to come out with a definition of where are my boundaries. So we, in this case, define 30% crossing is where the hard layer, diplo, porous layer, then that 30% is the cutoff line. And also we found that for front end, we looked at a lot of skulls and a lot of specimens at different locations. And then from front side, so this is the distribution. Definitely we saw statistically significant. Outer layer, there wasn't that much of a difference from front to side, but mid layer, these are 30% cutoff porosity, these boundaries. So there was a difference. So now we did compression experiment. These are tiny specimens we have using digital image correlation, speckling. We were getting stress. These are, and with cameras, we were measuring the strain distribution on the side of the specimen. And for human also, we did, these are a cubed specimen, both sides, one side and the other side. So, and these are the strain distribution. So this is really complex microstructure. So what we found was it simplifies us. It looks like layering. So I assume it is a layered structure. That gave us a way out in this complex structure. So if you look at the stress-strain relationship of these specimens, it's all over because microstructure is different from location to location. So you cannot use a stress-strain you obtain from you get cut a specimen and compress it. Each place will have a different stress-strain curve. So that is why you need to have the microstructure into the equation. So from this data, from each layer, we plot stress-strain. So these are stress-strain for each layer. So now for each layer, we can get a modulus. And then we have the porosity also. Now we can look at how to relate the initial modulus to porosity. So that's what we did. And the human also, we did the same thing. When somebody is doing this finite element analysis, person can assume this as a whole homogeneous structure or three-layer structure or multi-layer structure. So if you go to a multi-layer structure, it's very complex. Even meshing is complex. So what we decided was to give all this information and let the user decide what they want to use. This is we relate modulus as a function of bone volume fraction to the power k and E0. So it's represented by a power law. And because with Mill tensor measurements, pores were not anisotropic, we could ignore that part. So it boils down to, because I don't have time to go in detail into this. So we plot modulus as a function of bone volume fraction because we measured that. So and then this is my power law, and then I could get E0 and this power law. So when bone volume fraction 1 means that is modulus is E0, that is actual bone modulus. So it's a dense bone modulus. So I have that for mini pig, and then I have that for human, and then So human to mini pig to the age, all are embedded in this E0. So when you get older, your mineral content will be different, and then E0 will be different. So I have a power law, and then relating to the microstructure through bone volume fraction. This is really good. So also, this is modulus front versus side as a function of normalized thickness. And this is the key. Key is the modulus for 3-layered structure. I have that. And then composite structure if someone wants it. And then we use these moduli and did a finite element simulation of a specimen. And this is the digital image, DIC measurement on the side. So they are pretty good. So they are localizing at the center. And then modulus from the experiment, initial modulus and the simulation were almost the same. So we have for, how many more minutes? Okay. So we have for different pigs, how the modulus changes as a function of normalized depth. And for human also we have that. And then also we are working with MIT and Penn State using the meshing the whole bone and then doing finite element calculation and trying to get the E0 from this. And then does that having a layered structure versus non-layered structure, a homogenized structure, does that make a difference? This is one of my colleagues did this simulation, mini pig bumping into a plate. And this is the difference. So there's about 20 psi difference at this pressure. And for cellular level injury, this may be critical. Also, other thing we are doing is, these are ongoing work, we have developed high rate, what we call indentation experiments on skull caps. So using digital image correlation, where back surface of the skull, inner surface, we were measuring full field deformation. So this is the out of plane deformation in the back surface of the skull. And so our objective is to do a finite element simulation representing 3-layered structure and see whether we can predict this back surface displacement. So this is the simulation, and this is the actual measurement. So they're pretty reasonable. And then now we are working on trying to get this deviation. It is due to the crushing of the middle part. So that we are trying to incorporate. And I think, okay, so that's the kind of my presentation. I know I went really fast on skull. So if you have any more questions, and always you can call me and ask more questions. And
your work with the femurs, I assume, I've just made the assumption that the femurs were from male donors?
Yes.
Okay. With more females being involved in the military now, and their bone density changes as a function of their age more rapidly than men, how do you think that will affect your experimentation and your results?
This is the first study we did. Yeah, that's the direction. We need to add more younger and older female. We need to, and I'm also going through literature. Some of these may be already available in literature. because we have a now framework, power law, so I need to figure out how E0 varies as of sex and age, and all that come through the microstructure of the skull. So I need to go through literature and see. I'm sure some of these work here scattered around and see whether I can absorb something from the literature and get initial idea, and then look at the gaps and fill the gaps. So I'm sure there must be work on female with age, how the mineral density changes. So those are embedded in E0, so I have to split E0 into more detail to structural chemistry. Okay. Yeah, there's a lot of work to be done. This is initial stuff.
Bone structures are highly, I mean, as you indicated, it has microstructures. You can view that as composite materials that with embedded fibers, right? Your model seems to be based on assumption of continuum. Have you thought about, kind of utilize some kind of composite material formulation to, have you considered composite material type of formulation in your, material model?
When you say composite, it's
fiber composite, because those are a lot of behavior of bones.
Yeah, that is getting into another level of
details.
Substructure details.
Yeah.
So right now, I'm seeing porosity is a governing parameter. So what he was asking, when you get into that detail, just in the end, osteons consist of fibers. I think that's a long way for us to.
It's a long way to go.
Yeah, okay. And at least we are getting at this level and trying to incorporate. Yeah.
So just partially address, this gentleman's question about the female and osteoporosis for female. There was about decades of debate on how to define bone quality. Most people would consider bone density as the most important parameter to quantify bone quality. But a lot of study also has been reviewing that bone density may be only 50% of the bone quality. As you age, you lose that bone density. The other factor also play into it. One of them is the bone architecture. The cancellous bone architecture changes when you age. The third one may be the mixed. The constituent of collagen fiber versus the content of the organic material, which is when you age, your bone becomes more brittle because of you are losing some of the organic phase of the bone.
So I have already measured in some of these bones osteon distribution. I have quantified those things. But I couldn't, the three age groups, I could not correlate with osteon diameters. All I measured on all these three subjects, but I could not correlate. Yes, there is literature. I have a slide. I don't have it here. All the factors that affects also the collagen, how it with time elasticity get deteriorated. Yes, there is a lot of work, but in the Army we have to solve also problems. So they are not going to give us funding forever to explore all these.
In a human skull, there are the stitch marks where the different segments come together. And if one looks at that with an eyeball on the Weibull distribution and the fact that failure often occurs at the weakest point, i.e. the biggest defect, Do you see onset of that in the tests you've done, or is that far too much in the future?
So pig test, we have done things were failing on the suture lines. So we have selected pigs, so that is not the factor. So it is age is selected, so that is not the factor. And I would like to study this adhesion between the two layers, but that is going to change. Also, at the suture, the porosity is less. So that there are things to be studied, but right now, just looking at this is the, as far as I know, this detailed study so far. Mm-hmm. Yes, there are lots of things.
Yeah, I was wondering about on your tensile testing, the
bending test, bend test.
No, I'm talking about the stress strain graph that you showed, that you mentioned. You know, there are significant amount of variability. Did you like cut specimens from different locations of the bone throughout one, piece of bone, and then you looked at the stress strain graph, or you consistently look at one specific location of the same bone for different?
Yeah. So I had, for bent studies, I had 3 subjects, 4, and then I think it was 5 locations. Mm-hmm. So that wasn't, I could not correct. So because one human being at the middle, I have 4 specimens. Mm-hmm. Right? And then side, it wasn't, you could not do a study and not get into trouble with statisticians.
Right. Yes, okay? Sure.
So I could not do, so I assume it's the same material containing this same chemistry.
Yeah. But at the same time, it is concluded that, it seems that bone properties is in fact a local property, and it changes because of the porosity that you mentioned throughout the whole bone, right?
So let's say human eat the same thing, then chemistry is the same, then the architecture is going to be different.
Right.
So if over here versus over here, I had to assume somewhere it is not a factor for this kind of studies because it's getting into a spiral where you're going to go down and down, trying to.
So in the long range, are you then going to look at, designing phantom of the skull and the limbs, or designing body phantom and then looking at the impulse from the shock and how that transfers, once you understand all the issues with the bone structure? And I know, people in orthopedics, know about the bones very thoroughly and all the research. So what ultimately does this lead to? Protective measures?
There are the work I do have 2 paths. I'm trying to get material models to put into codes so that people like him can go and design things. Okay? And the other path is because I'm looking at the details of the microstructure. So that gives the people who are developing surrogate material. So we are also, he was involved in surrogate material. So we would like to have an anthropomorphic dummy so we don't have to have the cadavers being used or animal being used. So we are looking into surrogate material. I think APL has a big program on that. And we are also looking into it, human-like components. So that's the other goal. So that knowing the microstructure, I have a relationship where it should fracture, how it should bend, constitutive response, so then I can try to design some material which mimic that. So we are 3D printing some structures, and who are 3D printing skull structures. They're trying to match what I'm generating. Then I'm testing those also to see with the same methods, trying to whether they have done right, because there's a certain resolution you can get how you represent these pores when you're 3D printing.
We had a fascinating topic on bones. Any other questions you have for Tusit, please send him an email. If you need his email address, you can get it from me, or you can get it from the flyer. And thank you very much, Tusit, for a very, very interesting conversation. And thank you, people.